everybody, and welcome to this week's lecture on molecular biology. My name is Dr. Mark Temple, and I'm based on the Campbelltown campus, University of Western Sydney. So this week we're looking at Chapter 20, which is the regulation of gene expression in bacteria. Again, this comes from Molecular Biology by Cox et al. So this lecture isn't a substitute for reading the chapter, but hopefully it will focus you on some of the key points of the chapter that we think are important. So in this lecture today, we're going to be covering basically the control of gene expression in bacteria. And a case in point is the lactose operon. Okay, so we'll just discuss the lactose operon in some detail. Now, we've covered the lactose operon very briefly in the last lecture, so we're going to build up upon that content that we've already looked at. We're then going to extend looking at these bacterial operons and have a look at the tryptophan operon. Lactose is a sugar and tryptophan is an amino acid, and the cell has very different uses for each of these metabolites. So clearly the cell has very different responses to gene expression for the presence of these compounds. Okay, and we'll look at that in a bit of detail. And we'll kind of finish up the lecture by looking at the SOS response in E. coli. So effectively, we're looking at how a large number of genes can be regulated in response to a process. And typically, the SOS response is the activation of genes that play a role in the cellular response to DNA damage. And throughout this lecture, we'll be recapping on some principles that we've previously discussed in the previous lectures. So every now and again I'll show up a slide or two from the previous lecture, and then we'll embellish on that information in some way. So effectively what we've been looking at is the process of gene expression. And gene expression effectively refers to the information transfer from a DNA molecule through to the expression of a functional protein. And in this scheme, DNA contains the information that the cell requires to function. We then have an intermediate, which is the messenger RNA, and then from the messenger RNA we can then make the functional protein. So this is the functional molecular machinery, if you like, that performs cell function. As an aside, we've also looked at RNA as a catalytic activity in the cell as well, rather than just focusing on proteins. But for this general scheme, we tend to think of information going from the DNA through to the functional protein. And the main processes involved in this are transcription of the DNA to give a messenger RNA, and then we have the translation of the messenger RNA to give a protein. And we looked at protein synthesis in the first lecture, and we've looked at the flow of information in the second lecture. And what we're going to be focusing on this lecture is the control of transcription, which is an early stage in the processing of information. So in terms of gene expression, we say that a gene is expressed when the information encoded in that gene is transcribed and translated into a protein that's functional in the cell. Okay? And clearly we've discussed the concept that cells have many thousands of genes, and it's the coordinated regulation of these genes to produce proteins that are required at the right time and place that is the challenge for the cell. So clearly not all genes are expressed at any one time. That would be complete chaos to have everything being expressed simultaneously. And the examples that are given to make this point would be to think of, say, in higher eukaryotes, you'd look at a muscle cell that doesn't express the genes required that are particular to a neuron cell. and for instance eye cells express different genes from fat cells. So you've got all these different cell types that express different sets of genes. So clearly gene regulation is really important in this process of gene expression. In a bacterial system, we think about gene regulation in terms of responsive genes. So genes that respond to the presence or absence of a metabolite in their media or in the cell. And the typical examples of this would be to look at the Lac Operon, where the genes of the Lac Operon are induced by the presence of sugar, and we'll also look at amino acid operons, particularly the tryptophan operon, where the operon is inhibited by the presence of an amino acid. And we'll look at the rationale of why this is the case as we go through the material. So much of what we know about gene expression came initially from these pioneering work that was done in bacterial systems. So from the 1950s and 60s, people started to look at biochemistry and molecular biology in some detail and started to unravel the process of gene expression, DNA replications, and lots of other processes were really investigated in detail during this period. And if you remember from last week's lecture, we looked at 7 points that can be regulated in the flow of biological information. So looking at the points of regulation of gene expression, there are typically at least 7 points of regulation that the textbook tends to talk about. And in the first instance, we have gene transcription initiation, and this is what we'll be looking at today, and some of the pioneering work by Jacob and Monod to unravel gene regulation. Following this process, there are also opportunities at the stage of RNA processing to regulate expression, and typically there are introns that need to be removed in higher eukaryotes, and there's 5' caps and 3' tails, and there's various processing of the messenger RNA that's important. There's also RNA stability is an important factor in the control of gene expression, because effectively the more stable and long-lived the messenger RNA, then the more protein can be made from that message. And short-lived messenger RNAs are only available for a short period of time to make for protein synthesis. The stage of protein synthesis is also a point in the process that is subject to stringent regulation. And then once we've got a functional protein, we talked about protein modifications such as phosphorylation and acetylation and various processes. And also proteins can't be functional and said to be fully expressed until they're in the location and place in the cell where they're meant to be active, whether that be a membrane protein or a protein that's in the nucleus or some other protein. And also the last stage in gene expression is actually the degradation of proteins. And we talked about ubiquitination as a process of removing proteins from the cell once it's perceived that these proteins have fulfilled their function. So this is what we were discussing last lecture in terms of the flow of information. In this lecture, we're going to be focusing on particularly transcription initiation. And I should point out that when you're looking at gene expression in bacteria, it's a simpler system, and that one of the main reasons that gene expression is simplified in prokaryotes is that the DNA of prokaryotes is not in a nucleus. Okay, so in that case, you don't have a separation of the process of translation from the process of transcription. So whilst DNA is being transcribed, it can then begin to be translated straight away. Whereas in higher eukaryotes, transcription occurs in the nucleus, and then the messenger RNA has to be transported out of the nucleus into the cytosol, where it is then translated. So there's more opportunities to regulate this process because the two processes are separate in eukaryotes, whereas in prokaryotes like bacteria, these two processes are they happen sequentially, almost at the same time. So before transcription is finished, translation begins. And also in bacteria, the strength of the promoter plays a large role in the expression of a lot of genes. And in this case, a weak promoter is a promoter that doesn't have a high affinity for binding RNA polymerase, whereas a strong promoter has a strong affinity for binding RNA polymerase. Okay, so in the most basic sense, the simple affinity of the RNA polymerase for the promoter sequence of the gene has a lot to do with controlling the expression of that gene. So in bacteria, when we're talking about transcription, typically bacteria have operons, whereas in higher eukaryotes we don't have operons. Now, we've mentioned operons very briefly, and an operon is a collection of genes that are transcribed from a single promoter. So in the case of the Lac Operon, which we'll look at, there are 3 genes required for lactose metabolism. So it makes sense to have those 3 genes present in roughly equal amounts. And it makes sense to make more of those genes at the same time. And one way to achieve this is to have one promoter controlling the expression of 3 separate genes. And these separate genes in bacteria are clustered together in what we call an operon, where there's one promoter for 3 genes. Now, when this operon is transcribed, it gives rise to a single messenger RNA, and the messenger RNA is said to be polycistronic. I think that's historically genes were referred to as cistrons at some point. Polycistron just means many genes. So effectively it's a messenger RNA that codes for more than one gene, and that's said to be polycistronic. These polycistronic messenger RNAs contain multiple ribosome binding sites, and there's a single ribosome binding site prior to each open reading frame. So what this gives rise to is independent translation from the messenger RNA. And as I was saying just a short while ago, it's the affinity of the RNA polymerase to the promoter which, in the most basic sense, determines the control of gene expression. Clearly there are other factors that come into play, and we'll discuss these other factors, but if the RNA polymerase has poor affinity for the promoter, it doesn't bind strongly and it doesn't transcribe the gene. If the sequence of this promoter region here is a sequence that is strongly recognized by the RNA polymerase, then the gene is able to be transcribed at a higher efficiency. Okay, so it's the actual DNA sequence of the promoter that has a strong influence on the initiation of transcription. So different promoters have different strengths. Now, on top of this basic property of promoters, there are also other proteins that come into play in terms of gene regulation. And we've discussed these in the last lecture, and these typically are referred to as activators and repressors. So the activators and repressors confer additional levels of control on transcription. And in a very clever way, the activation of, or should I say the control of these activators and repressors is often tied in with the presence or absence of small molecules such as metabolites. And in the case of the Lac Operon, it's a byproduct of the sugar that controls the repressor. Okay? And likewise for the tryptophan operon, the tryptophan amino acid is also able to interact with the repressor to control the expression of that gene. These interactions are referred to as allosteric interactions because you have a small molecule which is simply binding and changing the conformation of that protein. And when you regulate a protein by changing its conformation through these ionic interactions and changes in structure of the protein, we refer to that as an allosteric interaction, which is in contrast to phosphorylation and acetylation and all of these covalent modifications to proteins. It's a different type of modification. So it's a ionic interaction of a metabolite to a protein rather than a covalent modification such as phosphorylation or acetylation. Here's our promoter, and here's our RNA polymerase that has some kind of affinity for that promoter, whether it be a strong affinity or weak affinity. But the actual ability of that RNA polymerase to bind to the promoter is interfered with by the presence of a repressor protein. So in this sense, the repressor protein stops the RNA polymerase from binding or extending from the promoter through the repressor binding site into the gene. Physically, it can't proceed. And the alternative to a repressor is an activator. And we discussed an activator, and we'll discuss repressors and activators during our discussion of the Lac Operon. But an activator is a protein which its binding to the promoter encourages the binding of the RNA polymerase. It encourages the RNA polymerase to bind more strongly and therefore regulate gene expression more strongly. So it's not just the affinity of the RNA polymerase to the promoter. This affinity can be modulated by repressors and activators to regulate gene transcription initiation and gene expression. So before we get into looking at the Lac Operon, I'd just like to quickly look at the kind of logic that one would expect of gene regulation. Okay, because if you understand what the cell's trying to achieve, and then you look at how the cells achieve that, it's a really interesting, problem solving that we can observe for this process. So clearly, E. coli are metabolizing various metabolites, amino acids and sugars. Okay, now we have two types of metabolism that the cell undergoes. Okay, we have catabolism, which is the breakdown of a molecule such as a sugar to release energy. Okay, so in terms of a sugar, the presence of a sugar would mean that the cell needs to express the genes to break it down. Okay, if the presence of the sugar, well, the bacteria goes, right, I need to express certain genes so that I can make use of that sugar to make energy. That's the logic. Okay, and with the other aspect of metabolism is anabolism. It's the building up of building blocks for and processes within the cell. So for instance, a cell may need to make amino acids so it can make proteins. Okay, so the cell has to make amino acids from various small metabolites. So if the amino acid is present, it doesn't need to make more. Therefore, the presence of the metabolite, meaning the amino acid, will repress the gene. Whereas in catabolism, the presence of the lactose induces the gene. Okay, so there's very different requirements for certain types of metabolites. Certain metabolites, you need to turn genes on to break them down, and other metabolites, if they're present, then there's no need to make them because it's a waste of energy. Okay, so we'll look at how the cell has overcome these two different issues in terms of gene regulation. So then, in summary, the bacteria cell needs to regulate the presence of various enzymes to make use of the nutrients that are available to it at any point in time. And to do this, it regulates its gene expression so that it can adapt to the environment which it finds itself. So to look at how E. coli regulates its genes, we're going to go back and look at some of the really interesting studies that were done on sugar metabolism in E. coli. Okay, so clearly E. coli can grow on different sugars, and it's very easy in the lab to grow bacteria on lactose or glucose or mannose or glycerol and all these different carbon sources. And a lot of early studies were doing this, but they were able to extend these studies to really get a glimpse of what's going on at the level of gene regulation. So I'm going to be talking about some early experiments done in the 1960s by Francis Jacob and Jacques Monod. Now, these two scientists are French scientists, so just go with the expression of their surnames as Jacob and Monod, even though it may not look like they're pronounced that way. These two scientists performed some very clever genetics experiments, and from these genetics experiments they were able to extrapolate to the level of gene regulation, and the conclusions they made about the regulation of the genes for lactose and how the lactose genes were regulated still ring true today. So it was a classic set of experiments that really, for the first time, looked at gene regulation and in an effective way. They won a Nobel Prize in 1965 for their work, and effectively their big contribution to science was that they discovered that some proteins' sole function is to regulate the expression of genes. Okay, so it wasn't understood prior to their work that there were such things as proteins that acted as transcription factors and activators and repressors. Now we take this for granted, and we don't really think about how we know these things exist in the very first instance. So we'll look at their experiments and the logic behind their experiments. So I just quickly did a screenshot, a Pubmed search for Jacob and Monod, and came up with a whole series of papers from the 1960s, and this is just the front of one of those papers. And you can see here they're looking at E. coli mutants, and they're looking at the regulation of the lactose system, and in this particular paper they're looking at some non-inducible mutants. So these were mutants that they couldn't switch on to upregulate the genes for lactose metabolisms. And they've had other mutants they discovered that couldn't be turned off, so that the genes were always on for to metabolize lactose even in the absence of lactose. So they basically performed mutational studies on E. coli, and then they selected for mutants that had different properties. Okay, now I don't want to get into too much of the detail of how this was done, but it's basic E. coli microbiology work. They expose the bacteria to a mutagen, and then they grow up those cells on probably glucose or something, and then they replicate them onto lactose, and then they look for ones that grow in the presence of one sugar but not the other, and then they can start to work out which mutants are unable to turn on the genes required. But let's not get into too much of that detail at that level. So lactose is a sugar that the bacteria can break down. I've drawn a little, I've got a figure here of lactose. It's basically two sugar units joined together by a glycosidic bond. So here we have our down, up, down sort of sugar, referring to the OHs. So this is our glucose, and here is a galactose. And if you remember the numbering of sugars, we go 1, 2, 3, 4, 1, 2, 3, 4, 5, 6. So we've got two 6-carbon sugars, and they're joined at a 1,4 position. So this is a 1,4 beta because it's in the beta position, a 1,4 beta glycosidic bond. So two sugars joined together by a glycosidic bond. Now these two sugars can be cleaved by an enzyme called beta galactosidase. So you've got a beta glycosidic bond that's cleaved by beta galactosidase. Now the beta galactosidase cleaves lactose into glucose and galactose. And glucose can feed directly into glycolysis, and galactose can be converted into a metabolite and also go into glycolysis. So glucose is the preferred carbon source for bacteria because it feeds straight into glycolysis without the need to turn on genes, and like it's constitutively expressed glycolysis, and glucose feeds directly in. So glucose is a preferred substrate, and when lactose is present in the cell, there are a set of genes that the bacteria can turn on to make use of this sugar. And effectively, lactose is milk sugar, so lactose is obviously present in milk. E. coli, they live in the gut of our intestines, and probably elsewhere. When E. coli come into contact with milk, they can then turn on the genes to metabolize lactose into these glucose and galactose so that it can derive energy from these sugars. But to do that, it cleaves the bond. Now, bacteria aren't always exposed to lactose, and in the absence of lactose, there's no need for the bacteria to turn on the genes for lactose metabolism. Okay, so again, it's that basic logic of why the genes are off in the first place, and then in the presence of lactose, they need to be turned on. Now, in the last lecture, we looked at the Lac Operon, and the textbook provided us with this schematic here of a promoter and containing activator binding sites and repressor binding sites. And the promoter is upstream from the gene, upstream meaning in the 5' direction, downstream meaning in the 3' direction. So this is the 3' end, the 5' end. So the promoter is upstream from the gene, immediately upstream from the gene. And this is the figure we looked at last week. Just for a bit of fun, I had a quick look in one of those papers and pulled out an early representation of the promoter from a science article. And again, here you can actually see, we're not looking at, don't worry about the base sequence, but you can see here that the tartar box in the Lac Operon is not a typical tartar box, and it's quite a weak tartar box, which, explains why the promoter is a fairly weak promoter. And they've also labeled some of the other elements of the promoter as well, such as a binding site for another protein here that we'll look at later. So effectively, the Lac Operon contains a weak promoter, but we know from looking at the studies of the bacteria in the presence and absence of lactose that these genes can be switched. This gene can be switched off so that it doesn't express any of the Lac Operon, and it can also be switched on, and it can be switched on to the extent that it's one of the most highly expressed genes in bacteria. So we have this weak promoter which can be in the off state or somehow converted into a very active on state. So here we have a schematic of the Lac Operon. Now the Lac Operon consists of 3 structural genes that code for enzymes or proteins that function in the cell. The Lac Operon also has a promoter shown here, and there's another region here referred to as Lac O, which is the operator binding site. Now up from the Lac Operon is another gene involved in lactose metabolism, and this is the gene that codes for the Lac repressor. Now the Lac Operon gives rise to 3 proteins, and they're shown here. The gene LAC-Z codes for the beta-galactosidase protein. The gene LAC-Y codes for a protein called galactoside permease, and the gene LAC-A codes for thiogalactoside transacetylase. Now, I've drawn the gene names here, but I'm actually drawing the protein, so don't get confused. I'm just relating the protein back to the gene by putting the name there. So, beta-galactosidase is the protein that catalyzes the cleavage of lactose into glucose and galactose. Okay, and then these sugars can be metabolized to produce ATP. Now, a galactoside permease, as its name implies, is a permease. So it's an integral membrane protein that allows the lactose to be transported across the plasma membrane into the cell. And the thiogalactoside transacetylase, not much is known about the acetylase, so we're not going to worry too much about the function. So in terms of looking at a cell here we have lactose outside the cell, and that lactose needs to get inside the cell. So effectively, the product of the LAC-Y gene, which is the galactoside permease, allows lactose to be transported into the cell. So this gives us intracellular lactose, and then the lactose needs to be converted particularly into glucose so that it can be metabolized. So the beta-galactosidase is the product of the LAC-Z gene, the LAC-Z gene, and this enzyme cleaves lactose into glucose and galactose. Now, during this metabolism, small amounts of another form of lactose called allolactose is also produced. Okay, so there were small amounts of allolactose also being produced in the conversion of lactose into glucose. And it's the allolactose that is registered by the Lac Operon, and it's the allolactose that actually regulates the operon. But allolactose can be thought of effectively as being lactose. I mean, whenever lactose is present, a small amount of allolactose will be present, and when lactose is absent, then allolactose will be absent. It is typically how it's described. So effectively, allolactose is going to be a main player in the regulation of the LAC repressor, but allolactose is effectively an indicator of the presence of lactose. And just lastly, we have the LAC-A gene, and again, people aren't really sure what that gene's doing at this stage. It's interesting, if you look at that paper I showed you from 1963 or something, then Jacob and Monod also put LAC-A ? We don't know what it does. And you think, it's 40 years later, 50 years later, and people are still saying, well, we're not really sure what LAC-A does. And I don't think anyone really cares, to be honest. It seems to be the way things go, because it's not involved directly in the in the gene regulation. So to study the activation of the Lac Operon needed a few tools. Okay, so as I was saying to you, allolactose is normally an inducer of the Lac Operon. Okay, and it's a product of lactose metabolism. Now, the presence of allolactose in the cell is quite hard to control. So if you want to study gene expression and regulation, you want to be able to fine-tune the switching on and off of that gene. So chemists came up with this compound, which is referred to as IPTG. Okay, it's got a long full name here, isopropyl thiogalactopyranoside, but basically it's IPTG. Right? This is an inducer in the same way that allolactose is an inducer. So you can see it's very similar to allolactose. It's similar enough to allolactose to be an inducer of the Lac Operon. And the reason molecular biologists or biochemists use IPTG to induce the Lac Operon is IPTG cannot be broken down by the cell. So if you want to add a fixed amount, a known amount of IPTG, and know that that's still going to be there in 10 minutes time inside the cell or 20 minutes time in the cell at a fixed concentration, then IPTG is really good. Right? Allolactose, it's very hard to control its metabolism inside the cell. IPTG is not metabolized by the cell, but it acts as a inducer of the Lac Operon. Another biochemical tool they needed to study the Lac Operon is when you turn on the genes for lactose degradation, how do you know you've turned them on? So you feed the cells a substrate. So xgal looks enough like lactose to be acted upon by the beta-galactosidase gene. Okay, when you cleave xgal, it gives rise to a blue chromophore. It gives rise to a blue color. Okay, so if beta-galactosidase, meaning the Lac Operon, is active, then this compound will be converted into a blue compound. So the combination of a non-metabolizable inducer and a substrate that didn't induce the operon but that could detect the activity of the operon, these two tools were enough to really study when and how these genes were turned on and off. So by growing bacteria colonies on a media that contained the xgal and IPTG, they were able to turn on the genes of the Lac Operon and detect the activity of the beta-galactosidase enzyme. And these tools allowed them to then really look at gene expression for this system. So Jacob and Monod performed a mutant screen of E. coli, and they identified various mutants. Okay, and then when they looked in a bit more detail, they identified the LAC-L and the LAC-O as two mutations that led to constitutive expression of the operon. Okay, so in the absence of lactose, the gene beta-galactosidase was turned on. So clearly they'd isolated some strains of bacteria where the gene regulation of the Lac Operon was broken, and then they could look at the detail of how it was broken, and then they could tweak those cells to restore activity, and then they could say, well, this is how it works, because we can now, fix these broken mutants, if you like. So their initial studies identified a range of mutants. Now, E. coli is a haploid, so there's one copy of the genes for the Lac Operon. It's not a diploid like higher eukaryotes. Now, the wild type has a phenotype that is on in the presence of the inducer. So the inducer turns these genes on. And in the absence of the inducer, these genes are off. And that's the wild type strain. Okay? Their mutant studies identified strains in which these genes were either on all the time or off all the time. So effectively, they identified a mutant in the LAC-L gene, which was constitutively on. Okay, so they identified a mutation in this gene here, so that these three genes here were being produced all the time, even in the absence of lactose, or in these experiments, in the absence of IPTG. They also identified another constitutively on mutation, and this one was in the LAC-O. So in the LAC-O region, they identified a mutant that was constitutively on in the absence of IPTG, the XGAL was cleaved into a blue product. They also identified a mutant, again, that mapped to the LAC-L gene, and in this mutant, the Lac Operon was constitutively off. So it had in the presence of IPTG, there was no blue color. Okay, so they were able to use the inducer and artificial substrate to see how the Lac Operon responded, and they were also able to map these mutations to particular genes and start to get a flavor of what was going on. So these are kind of raw genetics experiments, and Jacob and Monod were able to extend these experiments and then start to identify the fact that there was a repressor involved and that this repressor was controlling gene expression. So once these haploid mutants had been identified, Jacob and Monod constructed what we call merodiploid constructs. Okay, so these were strains of bacteria that were diploid for short regions of DNA. So effectively, they were diploid for the Lac Operon. So the way they did this was it's fairly simple really. They transformed bacteria with a plasmid that contained the Lac Operon. So you've got your genomic copy of the DNA, which I assume contained the mutation, and then they transformed the bacteria with a plasmid containing the wild type copy of the Lac Operon. So then they could have a look at the interplay between the two copies. So effectively, can the mutant LAC-L be restored by a wild type copy of the gene? Okay, and if it can, it says something about that gene product. And if it can't, it says something about that gene product as well. So they use this kind of analysis to understand what was happening with the gene products of the Lac Operon. So Jacob and Monod performed these merodiploid analysis, effectively making the bacteria diploid for the Lac Operon. So initially we were looking at a strain that was constitutively on, okay, and this was a haploid cell with a single mutation that mapped to the LAC-L gene. They then performed the merodiploid analysis on this by introducing a second copy of the Lac Operon, which was the wild type Lac Operon, into the cell. Now when you look at the combined phenotype now, you've got a mutant on one copy of the Lac-L and a wild type on the other. The resulting phenotype of this construct was wild type. Okay, so effectively, the presence of this gene here returned the cell to a wild type phenotype. Okay, so Lac-L wild type was able to overcome the mutation in the Lac-L gene, and the wild type product was able to return that cell into normal expression. So this product could turn this copy of the gene off. So given that the wild type Lac-L was able to rescue the mutant, they concluded that the Lac-L gene coded for a diffusible product that could act on either of the DNA molecules, not just the DNA molecule from which it was generated. So getting back to this diagram here, if you think about it, their conclusion, and we know this as a matter of fact now, but their conclusion was that this gene product, the wild type, codes for something which is able to diffuse and affect the other copy of the DNA that was mutant. All right, and effectively we know that today to be the Lac repressor. So if this mutant can't produce a functional Lac repressor, it can't repress this gene. If the wild type can produce a functional Lac repressor, it can diffuse across and bind to either copy of the gene, and it can then turn off the Lac Operon, and therefore in the absence of IPTG the repressor can function and you have a wild type phenotype. One of the other mutants they had identified was a mutant in the Lac O region, the operator here. So the phenotype of the haploid here is shown, and this strain was constitutively on. So in the presence of ITPG, it was on, but in the absence of the inducer, IPTG, it was still on. Okay, so couldn't turn this gene off. When you introduce a wild-type copy of the Lac-O gene, the combined phenotype of the two is still the same as the mutant phenotype. It's still constitutively on. So having a wild-type copy of Lac-O doesn't recover the mutant in the Lac-O. All right? So there isn't a diffusible product from here that can come and affect that. Okay? So again, I think what we know from the model these days is the Lac repressor can bind to the wild-type O to turn off that copy, but the Lac can't bind to this one because it's still mutant. So it can't turn off this. So the combined strain is still on, even though this is behaving normally. You can't recover the mutant in the Lac operator. Okay? And Jacob and Monod, they were constructing the model and they were trying to work out what was going on. And in contrast to the Lac-L mutant they'd looked at, the wild-type copy of the Lac-O was not capable of rescuing the defect in Lac-O. So the Lac-O did not code for a diffusible substance. Okay? So from these kind of experiments, they did a whole bunch of these experiments, and they were able to identify two main elements controlling gene expression. The first was that the Lac repressor was encoded by the Lac-L gene, and that the Lac-O was actually a binding site for the Lac repressor. Okay? So the Lac-O did not code for a gene, didn't code for a product, but it was a binding site of the repressor. And in the absence of lactose, the genes were not transcribed because the Lac repressor binds to the operator in the normal wild-type cell. So before Jacob and Monod did these experiments, the concept of a protein acting as a repressor or activator for a gene, it didn't exist. They didn't think of gene regulation in those terms. It wasn't known. I mean, people might suspect, but they hadn't demonstrated that that was the case. And also, if you look at Jacob and Monod's papers, I think they imply that RNA rather than protein was probably what the repressor was. And clearly they were wrong in this instance because we know the Lac repressor is a protein, but it just gives you an insight into how much was known and how they were struggling to put the pieces of the jigsaw puzzle together to really understand what was happening. So we know that the Lac-L then codes for a protein, okay? And that protein we know now is the Lac repressor. So we have a promoter leading to expression of Lac-L, which is the Lac repressor. Now this is not induced by IPTG, it's just being produced at low levels in the cell. So the cell produces the repressor. Now the repressor binds to a region of the promoter of the Lac Operon called the operator. Now the operator is between the promoter and the structural genes. So where the RNA polymerase binds, it then the polymerase needs to read through the operator to get to the genes. And if the repressor is there, then the RNA polymerase cannot transcribe the genes. So effectively the Lac repressor can turn off the operator, and effectively here, this is a little schematic from last week that was referring to the generic principle of repressors. So in this scheme here, if we were referring to the Lac Operon, then this red blob here would be the Lac repressor. Okay, and I think what I was talking about last week was that the presence of the repressor stops the binding of the RNA polymerase, or stops the RNA polymerase from reading into the genes, and you have no transcription. Once the repressor dissociates from the promoter region, the RNA polymerase is able to initiate transcription and then produce a polycistronic messenger RNA which has the Lac genes. So in the presence of lactose, or in their experiments IPTG, then the Lac repressor is unable to bind. Allosterically, it's changed structure and it no longer has high affinity for DNA. And that allows the polymerase to bind and effectively turns on the Lac Operon to produce the BGAL and the permease. So last week we looked at some really nice X-ray crystallography images of DNA binding proteins binding to DNA. And we looked at leucine zippers and helix turn helix and some other motifs that occur commonly in DNA binding proteins. And we also discussed that the binding site for DNA binding proteins is often a inverted repeat sequence. So there's a short sequence of bases, and then there's a couple of bases in between, and then there's that short sequence repeats, and the binding site for transcription factors is often an inverted repeat. Now, when you look at the Lac repressor in a bit more detail, it's actually a tetramer, not just a dimer. So it can function as a dimer, but then those two dimers come together to form a tetramer. Okay, and we also looked last week at a looping region of DNA at the Lac Operon. So we'll have a look at that in a bit more detail. So recall that we looked at these helix turn helix motifs of proteins, and the Lac repressor contains the helix turn helix motif. Okay, which is why I'm talking about it. So the regulation of the Lac repressor is a classic example of allosteric regulation, where a small metabolite binds to the protein, changes the conformation of the protein to make it bind more strongly or more weakly, depending on the case, to DNA. Right? So in the case of the Lac repressor, adjacent to the helix turn helix region is another helix, which is referred to as the hinge. So it's a partial loose bit of loop which can form a helix. Okay, and I'll show you a picture of this. Inducer binding, so the IPTG or allolactose binding to the Lac repressor, causes the disordered hinge region to form a helix which binds very strongly to DNA. So just a quick refresher on helix turn helix motifs. Here's our bit of DNA here, and here's a generic domain of a protein which is a helix turn helix. The Lac repressor contains a helix turn helix. So if you look here, this is a helix turn helix here, and this is one copy of the dimer, and this is the helix turn helix of another copy of the dimer. And this is only showing the DNA binding domain of the Lac repressor. The Lac repressor is much bigger than this, but it's just showing the DNA binding portion. So the helix turn helix is binding to the inverted repeat. The protein's a dimer, so it fulfills all those things that we know about these DNA binding proteins. But what's interesting, that there's this loose hinge region here. It's like this region here and I guess there's a copy of that region on the other protein as well, they can, upon allosteric binding of the of allolactose, changes the structure. So there's a little animation here. So here's our helix to helix motif of each of the proteins, and here is that hinge region, and upon binding the allolactose, it changes so that this unstructured region becomes structured. Okay, so in this form here, you can see that the hinge region forms this alpha helix, and the same with the other copy here. So once the allolactose binds, it changes the DNA binding domain of the Lac repressor, and it binds so much more tightly to the DNA, and it actually causes the DNA to bend. You can see here the DNA is bent, whereas in this form, without the inducer, these helices haven't formed and the DNA is not bent. So it's this binding and bending of the DNA that then allow the Lac repressor to loop out the intervening DNA. Okay, so now also since Jacob and Monod, people have worked out that there's not just one operator site. There are actually about 3 operator sites. So there's the O1 site, which is the operator site that was identified by Jacob and Monod, and then there's some other operator sites referred to as O2 and O3, which are positioned in different places close to the first operator. So again, this is just a slightly more detailed view of the Lac Operon. Here's the initial operator that we've been looking at, where we know the Lac repressor binds. There's also another operator here and another operator here. Each of these 3 operators has an inverted repeat for the binding of the Lac repressor. So a dimer can bind here, and another dimer, for instance, can bind here, and then those 2 dimers bend the DNA and come together to form a tetramer. So effectively, one dimer binds to one of the operators, another dimer binds to either the O2 or the O3, one of the other operators, and then these 2 dimers come together to form a tetramer, and this formation enables the looping out of the DNA at the regulatory site to turn off the promoter. So in the last lecture we looked at this kind of structure. This is just a bit more detail. So here we have, in this instance, the Lac repressor dimer bound to O1 and another Lac repressor dimer bound to O3, and then these 2 dimers come together to form a tetramer. It also causes bending of the DNA, and it causes a looping of the promoter to turn off the Lac Operon. And again, this is just the textbook showing you way too much detail, a really big X-ray crystallography structure of a Lac repressor tetramer bound to a bit of 2 bits of DNA, forming these regions here. So these are the operator regions here. So when you go back to Jacob and Monod and their original experiments, some of the things they did during their experimentation was popularize or develop these techniques which are still used today in molecular biology. And I'll just quickly mention a couple of these techniques and show you quickly how they function. So these techniques are gel shift assays and footprinting, or protein DNA footprinting. Okay, so both of these techniques have a similarity. The first is, to run these techniques, you need to get a piece of DNA, a PCR product, or they would have used sort of restriction digest bits of DNA probably. And then these bits of DNA, such as the Lac Operon, for instance, a bit of DNA representing the Lac Operon, you would label at one end. They would have used radio label. Nowadays we would use a fluorescent label. So effectively, you label up your piece of DNA that's of interest to you. You then get a extract of proteins, and you bind your protein, this blue bit, purple bit here, to the DNA. And then you have a bit of DNA plus or minus a bound protein, and the DNA can be visualized. So the gel shift assay is the most straightforward of these two assays. So effectively the gel shift assay uses an agarose gel or a polyacrylamide gel, some sort of matrix to separate out the DNA. You run your DNA against a marker and you know that's where your DNA runs. So effectively you electrophorease your bit of labeled DNA and it has a certain position in the gel. You then get your cell extract, bind it to your bit of DNA, and you see whether there's a specific binding of a protein to that bit of DNA. So if you've got a bit of protein that's specifically bound to the DNA, this piece of DNA doesn't migrate like the unbound bit of DNA, but its migration is retarded. Okay, so it's also referred to as a gel retardation assay or gel shift assay. Shift meaning it's shifted from here to here. A retardation assay meaning it's been retarded, it can't go all the way down. Same name, different names for the same experiment. And if you increase the amount of protein across these 7 samples here, as you add more and more protein, more of this label accumulates in the bound position. And if you add more and more protein, then the amount of free DNA gets less and less until there is no more free DNA. It's all bound by the protein. So using these relatively straightforward experiments, they were able to establish the binding of the protein to the DNA using some fairly simple molecular biology techniques. Another technique which is fairly similar is called a footprinting assay. And again, it's quite similar. You get your bit of DNA of interest, you label it, and then you bind protein to your bit of DNA, and then you try and work out where on the sequence it's bound. So the gel shift assay told you it was bound. The footprinting assay tells you where it's bound. So in the footprinting assay, you do a bit of extra work. You take your protein bound to a bit of DNA, and then you use another reagent that cleaves the DNA. Okay, and if there's no protein bound, the DNA is equally cleaved into a range of fragments. Okay, and then you cleave the DNA into fragments, and you run those fragments on a gel, and depending on where it's cleaved, you get a different size product. Whilst you're doing this, you then cleave your piece of DNA that's got a protein bound, and where the protein is bound, it can't be cleaved. Okay, so you don't get any size products in this region here. Okay, and that's called a footprint. So when you run these products on a gel, you get a bunch of small fragments here, you get a bunch of large fragments here, but you don't get any medium-sized fragments because the protein protected that DNA from being digested. So what you get when you run your sample that was bound to protein to your DNA sample that wasn't bound to protein, and both of these have been digested, is you get an absence of bands. And that absence of bands here is a footprint. Okay, think of it as putting a footprint in a sand. It tells you something was bound there, something was there, and it's left a footprint. And an actual gel shown from the textbook. Here we have a sequencing lane. This is just showing the presence of C residues on the template. Here we have a piece of DNA with no protein bound. When this piece of DNA was digested, you get a series of bands across the entire piece of DNA. This piece of DNA was bound to a protein. In this case, it was RNA polymerase. And the binding of the polymerase stops these regions of DNA from being damaged by whatever damaging compound they added. And therefore, you get this big footprint here that says at these exact bases the protein was bound. So this is the sequence, this is the cleavage plus protein, and these big footprints here are the footprints of where the protein was bound to the DNA. So these are great techniques, and they're still used in labs today. So getting back to the Lac Operon, we've looked at the repression of the Lac Operon. Okay, so when there's no lactose present, the genes are off. Clearly, when the cell's exposed to lactose, the lactose needs to get into the cell. The lactose can't get into the cell unless the permease is present. Okay, so even with this efficient system of the Lac repressor switching off the Lac Operon, there is a small amount of expression of the Lac genes. And that small amount of expression is the cell kind of hedging its bets, producing a small amount, small energy cost, puts a bit of the permease in the cell membrane so that when lactose is present, it can actually bring it into the cell and then activate the genes properly by turning off the repressor. So you have to have a small basal level of expression of these genes, because otherwise lactose can't get into the cell. Okay, so essential to this model is to have a little bit of leaky promoter producing a very small amount of these genes, particularly the permease, to bring the lactose into the cell so they can be converted to allolactose to remove the repressor. So to this point, we've discussed the induction of the Lac Operon in the absence of glucose. Okay, and we know from sort of just an arm-waving rational point of view that glucose is a preferred substrate, and that when glucose is present, even if lactose is present, the cell will preferentially use glucose, not lactose. So somehow there has to be a mechanism where glucose turns off the Lac Operon. And when glucose is absent, then the Lac Operon can be more active. So the reason that glucose is a preferred energy source is that it can feed directly into glycolysis. And again, from last week's lecture, we talked about this, and I'll just refresh your memories on this. The presence of glucose affects the Lac Operon. Now, it's not directly the glucose, but it's another metabolite which is a signal of the glucose, if you like. So, effectively, low levels of glucose in the cell lead to high levels of a metabolite called cyclic AMP. So this small c, large AMP is cyclic AMP. Okay. Now, cyclic AMP is a small metabolite that is an effector for a protein called the cyclic AMP receptor protein, which is also referred to as catabolite activator protein. Now, the textbook mainly refers to it as cyclic AMP receptor protein, so CRP. So the low levels of glucose lead to high levels of cyclic AMP. The high levels of cyclic AMP is an effector for this receptor protein. And we'll see how this scheme fits in with regulation of the Lac Operon. So here's a animation to show the allosteric activation of the CRP to make it have a higher affinity to bind DNA. So effectively, in the absence of glucose, which is the conditions of Jacob-Monod's experiments, then the CRP was activated and was bound to DNA. Now, this protein increases the affinity of RNA polymerase for the promoter. So in the presence of this structural form of the CRP, the RNA polymerase binds to the promoter with high affinity. So when the repressor is removed, this thing can then transcribe the genes at a high rate. So effectively, the lactose binds to the repressor, removes the repressor, and because this is present, it makes the affinity of the RNA polymerase for the promoter much stronger, and this leads to high-level transcription in the presence of lactose and the absence of glucose, because low glucose, high cyclic AMP. High cyclic AMP, this protein, this activator binds and increases the affinity of the RNA polymerase. So obviously this came about because people started to look at the Lac repressor in the presence of glucose. And in the presence of glucose, the cyclic AMP levels are low. So in the presence of glucose, the RNA polymerase affinity for the promoter is weak. And therefore, even in the presence of allolactose, the RNA polymerase is fairly feeble, if you like, at producing and binding to the promoter. At some point it will bind to the promoter and produce low-level transcription of the lactose operon genes, but this is because glucose is present, and in the absence of glucose it's a much higher activation of the Lac Operon. So this is just showing the structure of the cyclic AMP receptor protein. Again, it's showing very clearly that it's a DNA binding protein. It's showing very clearly that it's operating as a dimer, binding to these invert repeats. It's showing very clearly that it's got two binding sites for cyclic AMP, one binding site in each of the proteins of the dimer. And it's also showing that it's got another region here, which is the region of the protein that binds and encourages the RNA polymerase to bind more strongly to the promoter. So it's the binding of this protein to the promoter that makes the RNA polymerase have a higher affinity to the promoter. Because remember, the promoter has a fairly weak tartar box. Okay, so this is just a summary of the operation of the Lac Operon. Under conditions of glucose being high, then we have cyclic AMP low, so we're not showing the binding here of the CRP protein, it's absent. And lactose is absent, therefore the repressor is bound. And under these conditions, the wild type is off. When lactose is present, then allolactose is present because lactose is present, then effectively the repressor binding is relieved, and therefore the polymerase can bind weakly and express the genes at a low level. When glucose is low and lactose is absent, there are high levels of cyclic AMP. The cyclic AMP binds to the receptor protein here, which can then bind to its site here, and effectively the repressor is still present, so you've got no gene expression, but when the repressor is absent, the polymerase can bind more tightly, as shown in this one. So high levels of cyclic AMP, meaning low glucose, strong binding of the polymerase because the protein encourages the polymerase binding, and therefore you get high level expression of the Lac Operon. So the cyclic AMP repressor protein doesn't only affect expression of the Lac Operon. Okay, it's involved in the activation of other operons, in particularly the arabinose, which is a sugar. The arabinose operon is regulated by the cyclic AMP receptor protein, and also the galactose operon is regulated by this same protein. So in this way, glucose, the preferred energy source, can regulate the expression of other genes for other sugars. Okay, so when glucose is present, these other genes are downregulated. When glucose is absent and arabinose is present, or when glucose is absent and galactose is present, then the arabinose or the galactose operon can be activated in the same way that the Lac Operon is partly regulated by the presence or absence of glucose. Okay, so we're now going to change tack and look at another operon, and our understanding of the Lac Operon certainly helps our understanding of the trip operon. The trip operon, now tryptophan, as you may recall, tryptophan is an amino acid. So when tryptophan is present, you don't want to make more tryptophan, okay? But when tryptophan is absent, you want to make more tryptophan. And this is the opposite of lactose, okay? So the control of the tryptophan operon follows a different logic when we think about it, because we're dealing with a very different metabolite. Here we're looking at a metabolite that's involved in anabolic metabolism, the building up of things. Making proteins requires tryptophan. When tryptophan's absent, you need to make tryptophan. Therefore, you need to turn genes on when the tryptophan's not there to make more. So these amino acid operons are repressed when existing supplies of the amino acid are present. And when more amino acids are needed, then you need to actively transcribe the genes to make that amino acid. Again, the trip operon has a repressor protein that is allosterically regulated by the metabolite, in this case tryptophan. So the trip repressor is a homodimer, and tryptophan acts as a small molecule effector for the trip repressor. So when tryptophan is present, the repressor is active. So here's a little basic animation taken from the textbook. Here is the trip operon. There are 5 genes in the trip operon, trip E, D, C, B, and A. I don't know why they're in reverse order to A, B, C, D, and E, but that's just the way they are. So here are the 5 genes of the tryptophan operon. We have an operator here, which is the binding site of the repressor. The repressor is a dimer, and here's the promoter. In the absence of tryptophan, the repressor is not active. So in the absence of tryptophan, the genes are on. The presence of tryptophan causes the repressor to bind and turn the genes off. So the presence of tryptophan causes the repressor to turn off the genes, because you don't want to make tryptophan if tryptophan is present. If tryptophan is present, then you can make proteins from that amino acid. There's no need to make more tryptophan. Again, here's one of these X-ray crystallography diagrams showing that the tryptophan repressor is strongly bound to DNA in the presence of the of tryptophan. Okay. So when we looked at the Lac repressor, it was the Lac repressor bound in the absence of allolactose. When you look at the trip repressor, the repressor binds in the presence of tryptophan. Okay, so there's the cells develop this machinery that responds differently to the presence of the metabolite. The Lac Operon is catabolic, and the presence of the metabolite turns the genes on so it can be broken down. Tryptophan is an amino acid. We make amino acids, so the presence of the amino acid signals that we don't need to make any more. Therefore, the presence of the amino acid turns off the promoter. It makes a lot of sense logically, even though logic often doesn't apply to cells. In this case, it does. So we can quite logically predict whether the metabolite will activate or not the gene based on whether it's catabolic or anabolic. Now, the TRP repressor is also interesting because it has another mechanism of regulation. And again, this is a mechanism of regulation that occurs in quite a few amino acid biosynthesis operons. And this method of regulation is called attenuation. Okay, so effectively, once transcription has begun, attenuation is a secondary regulation point that can stop transcription. Okay, so we've had the polymerase bind, we've had the repressor on or off, and we've got the transcription has started. Now, once transcription has started for the TRP operon, there's another regulation point that can stop transcription. Okay, and this mechanism is called attenuation. Now, attenuation only really makes sense in bacteria, because in bacteria, translation follows transcription immediately. So as the gene is being transcribed into a messenger RNA, the ribosome binds and starts to translate it, okay? Because they're not separated by a nuclear envelope, okay? So attenuation relies on the close coupling of transcription and translation. And I've sort of created an animation that covers this process. And so what we have here is the blue double line here is our double-stranded DNA. The RNA polymerase is bound. Here's our open conformation. And this green line here is the messenger RNA. So this is showing that in prokaryotes, the ribosome is bound to the messenger RNA as the messenger RNA is being transcribed. So the polymerase has to run a lot further to transcribe the entire messenger RNA. But as it's being transcribed, translation begins. So in this scheme here, we have the polymerase transcribing the DNA into a messenger RNA, and as the messenger RNA is being made, it's being translated into a protein. And this is called coupled, a coupling of translation to transcription. So the cell, bacteria cells, have used the fact that there's a coupling between these two processes of translation and transcription, so that the process of the messenger RNA is able to form a structure that stops transcription. And you'll see that in the next few slides. So here's our promoter region here. Here's the operator. So this is what we understand from biosynthetic, from different operons. What's different in the amino acid operons, tryptophan operon, is what's called a leader sequence. So transcription starts around here, and then there's a leader sequence before all of these structural genes to make the enzymes required to make tryptophan. And it's what happens during the transcription of the leader into a messenger RNA that can attenuate and stop the transcription of these genes. So there's something special about this leader sequence that can respond to the amount of tryptophan present, and then it can regulate transcription based on the amount of tryptophan present. Okay, so preceding the codons of the TRP operon is this leader sequence, and the leader sequence consists of 4 regulatory regions labeled 1, 2, 3, and 4. Okay, so these yellow sequences here are the 4 leader sequences. Okay, these leader sequences contain are GC rich and they can form hairpin loop structures between themselves. So region 2 can form a hairpin with region 3, or region 3 can form a hairpin with region 4. Okay, and depending on which hairpin forms, you either allow transcription or stop transcription. Okay, the other important bit of information is that there's a leader sequence here which contains 2 codons here for tryptophan. So don't forget, this is the Trip operon, and if tryptophan is present, then you can easily find charged tryptophans, which are tryptophans attached to transfer RNA, and therefore the ribosome can translate through this region quickly and easily if tryptophan is present. If tryptophan is absent, then it's very difficult for the RNA polymerase to read through these trip codons because there is no tryptophan present. There's no charged tryptophan tRNAs, and therefore translation will pause here. Okay, so the process of translation uses charged tryptophan, and then if everything is satisfied and okay, so in this way the process of translation is sensitive to the amount of tryptophan that's present in the cell. And this is just showing that these leader sequence here, these 4 regulatory regions in the leader sequence, are part of the operon. So here are the transcription, here are the structural genes here, and then upstream of these is the leader sequence with these 4 regulatory regions, and then way up here is the promoter, the operator binding site. So we're assuming that transcription has begun, and it's now looking at the process of producing a messenger RNA with these regions. So, like I said, there are 4 of these regulatory regions. We've got 1, 2, 3, and 4. So this is 1, and then 2, 3, and 4. These regulatory regions can bind to each other because they're GC rich and they are complementary base sequences to each other. So which stem loop structure forms determines whether or not transcription will proceed. Now, sequences 3 and 4, which is this one and this one, can form a hairpin structure. So this is showing the leader sequence with a hairpin between regions 3 and 4, and here's the base pairing between the GC-rich base pairs. Now, important bit of information is that just up from this region 4 is a series of U bases, uracil bases, okay? So the hairpin structure here, followed by the uracil residues, is a termination structure, a terminator, okay, of transcription. And I'll show that in a couple of slides' time. Now, the other structure that can form is a hairpin between regions 2 and 3. Now, the formation of this hairpin structure here doesn't act as a terminator, because directly here there are no uracil residues here, so it doesn't fit our description or our sort of understanding what a terminator is. Okay, so something happens when the ribosome is translating here, which affects whether the hairpin forms between 2 and 3 or 3 and 4. Clearly, if 3 is bound to 2, then 3 can't bound to 4, and therefore you can't get this structure. Okay, you either get one or the other. So the regulatory sequence 1 also has codes for a very small protein, and that protein contains 2 tryptophan residues. Now, as sequence 1 is translated, it determines whether sequence 3 will base pair with 2 or 4. and I'll show that in these diagrams. We call that sequence 1 encodes a small peptide with 2 tryptophan residues. The peptide it encodes is only a 14 residue peptide, and it's not a functional protein, but it's a little peptide that requires the presence of tryptophan. Okay, so immediately after this region of the messenger RNA is transcribed, it is translated. Okay, and as it's translated, the ribosome looks for these tryptophan, charged tryptophans it needs to read through the trip codons. So here there's an animation just showing the coupled translation and transcription. This time we're looking specifically at this leader sequence of the trip operon. So here's our ribosome attached to the messenger RNA that's been transcribed. Now, under conditions where concentrations of tryptophan are high, these trip residues here can easily be translated. Okay, so the ribosome can read through these trip residues because it has charged trip tRNAs that can come in and make the protein. So effectively, the ribosome can read through to the stop codon of this very small gene here, and it causes it to pause and overlap region 2. Therefore, a hairpin can form between regions 3 and 4 as the messenger RNA is being transcribed. Okay? As soon as this region, this hairpin here forms with this rich uracil sequence here, this is a terminator structure. So this hairpin here will form, and it will butt off the, it will affect the binding of the RNA polymerase, and also the uracil residues weakly hydrogen bond here to the DNA, and therefore it's able to effectively terminate transcription. So given the fact that the ribosome can read through these tryptophan codons, the sequence 2 is then quickly covered by the ribosome as the rest of the messenger RNA is being translated, or transcribed, sorry, allows this terminator structure to form, which then halts transcription. So it's the availability of tryptophan allows the polymerase to read through these trip codons to the ribosome sits on region 2 and allows region 3 and 4 to form this terminator, which is complemented by these uracil residues. So again, from the textbook, just to summarize what I've just said to you, when trip levels are high, the charged tryptophan tRNA levels are also high. Okay, this allows the ribosome to translate through the trip codons, and therefore the ribosome covers sequence 2. So as regions 3 and 4 are transcribed, it allows a hairpin to form, and then this hairpin, as it forms with the poly-U sequence as well, it acts as a terminator to halt transcription. Okay, and don't forget, the structural genes haven't yet been transcribed. Okay, the leader sequence is before the structural genes of the trip operon. Now, when regions of tryptophan, when concentration of tryptophan is low, the ribosome reads through these up to these trip codons, and it can't proceed. Okay, there's not enough charged tRNAs to allow the ribosome to proceed down. So the ribosome doesn't read down to cover region 2. Therefore, as region 3 is transcribed, a hairpin can form between 2 and 3. Okay, 2 and 3, this hairpin here doesn't have this poly-U sequence here. It doesn't act as a terminator of transcription. Therefore, transcription proceeds, and then the polymerase keeps transcribing, and it will transcribe those 5 genes required to make more tryptophan. Okay, so effectively the translation stalls because there's low tryptophan. Sequence 2 is available because the ribosome is not sitting on it to base pair with region 3, and therefore the entire operon can be transcribed, and therefore the ribosome can continue and translate the structural genes into the proteins required to make more tryptophan. To reiterate what I just said, when concentrations of tryptophan are low, the ribosome stalls at those 2 trip codons in that tiny little gene at the beginning of the leader, and therefore sequence 2 remains free and not read by the ribosome, so region 2 can hairpin and bind to region 3, and then this prevents the formation of the terminator. Okay, now terminator is the base pairing of region 3 to region 4. That can't happen once the hairpin's formed between 2 and 3, and therefore transcription proceeds. Now this process of attenuation isn't unique to the trip operon, and if you look at the operons of phenylalanine, leucine and histidine, then the leader sequences of these operons also have multiple phenyl residues in the operon, has multiple lysine residues in leucine residues in the leucine operon, and multiple histidine residues in the his operon. So clearly this process of attenuation is occurring in these amino acid biosynthetic operons as well. Alright, so I'll finish off this lecture with a fairly short section just looking at these SOS response genes. Okay, so this is looking at a concept we covered briefly in the last lecture, and this is this idea of a bunch of genes separated around the genome are coordinately regulated by the same repressor protein. So the SOS response is the cellular response to DNA damage. So when DNA is damaged, you get the activation of a bunch of genes involved in helping DNA repair. So effectively many genes that are required for a particular function are regulated together. And the example we're looking at here is a cell response to DNA damage. So when you have excessive breakage of the bacterial chromosome, certain proteins recognize that damage and bind to the damage, and somehow these proteins can then affect the regulation of genes required to respond to that damage. So this response is known as the SOS response, and there are 2 key regulators that we need to think about. We have the REC-A protein and the LEX-A protein. Now LEX-A is a repressor bound to the genes involved in DNA repair, and the REC-A protein is the protein that binds to DNA damage. Okay? So as I just said, the LEX-A protein is a repressor that binds to the promoter of these SOS genes, so these genes involved in DNA synthesis and repair. So the induction of the SOS genes requires removal of this repressor. Now, the removal of the repressor in this case isn't through allosteric interaction with a small metabolite. Okay? When you damage DNA, there is no small metabolite to signal to the repressor. Okay? So we'll look at the interesting way in which this system works so that the repressor is removed upon DNA damage. And if you read this dot point here, I'm telling you that the LEX-A repressor is removed through self-cleavage. Okay, so the LEX-A repressor is actually degraded and therefore can no longer act as a repressor. So this system works because the LEX-A repressor binding site, the protein is continually binding and leaving and binding and leaving, and there's enough copies of this repressor to continually have the genes off. Okay, so you've got this cycling of repressor binding on and off the promoter, but effectively keeping the promoter in the off state. So I've done a little animation here to sort of show that this repressor protein is cycling on and off from the promoter, but effectively the gene is still off. Now somewhere else in the genome, as shown up here, some UV damage or some chemical carcinogen or something's happened to damage the DNA. So we've got some single-stranded DNA present as a measure of DNA damage. This other protein, the REC-A, it binds to damaged DNA. So you get this REC-A protein binding here. Now, as the LEX-A protein, the repressor, is binding on and off its promoter sequence, it's also binding loosely to other bits of DNA. It's floating around the cell, okay? It's not fixed to the repressor. So the LEX-A protein can come into contact with the REC-A bound to damaged DNA. So every now and again, these LEX-A proteins bump into the REC-A, and that causes the REC-A protein to induce its own cleavage. So effectively, this structure here causes the free version of the repressor to be degraded. So over time, these repressors, rather than binding to only the promoter, they get degraded because there's this on-off, movement of these repressor molecules. And as the repressor gets degraded because of the DNA damage, then the promoter of these genes are then the repression is relieved because the repressor has been broken down, and therefore these SOS genes can be activated. And I showed a figure very much like this last week. I've just embellished it a little to reflect what's happening at these SOS genes. So effectively, these repressors are leaving and binding to the different promoters. At some point, the DNA damage causes the repressors to self-cleave, and therefore it removes the repressors from the promoter. And simultaneously, a whole bunch of genes that share function have been activated in response to the environment. In this case, it was DNA damage causing the breakdown of the repressor to turn on genes required to fix and help the cell respond to the damaged DNA. So in this SOS response, the REC-A was activated by binding to damaged DNA, and then the REC-A gene induced cleavage of these LEX repressors. And this system worked because the LEX repressors, the LEX-A repressor molecules, were constantly cycling on and off from the promoter. So when they're off the promoter, they sort of clearly bounce around the cell and bind to come into contact with these active REC-A molecules on the single-stranded DNA, and that causes the degradation of the repressor. So as the repressor is digested, it leads to it relieves the repression of those SOS genes. So just to summarize some of the key points on this lecture in relation to the dot points that are in the textbook, we talked about the Lac repressor, and this was the studies by Jacob and Monod that first understood gene regulation in a really practical way, and they identified proteins whose function was to bind and repress or regulate the gene expression and to regulate transcription. And this was the first time that people had identified proteins that weren't just enzymes or structural proteins, but they were DNA binding proteins that regulated gene expression. We also talked briefly about catabolite repression. In those early studies on the Lac Operon done in the absence of glucose, then there were high levels of cyclic AMP, and this caused these positive regulators to bind to the promoter to encourage the RNA polymerase to transcribe at a high rate. And catabolite repression refers to when glucose is present, the cyclic AMP is absent, and therefore you don't get this activation and this strong binding of the RNA polymerase. And this is a way of coordinating sugar metabolism, so the preferred sugar is metabolized even in the presence of another sugar such as lactose. The cell will still preferentially use the glucose and not activate the lactose genes until the catabolite repression is over, until all the glucose is consumed. We also talked about attenuation, and in relation to the operons involved in amino acid synthesis. And effectively, this process takes advantage of the fact that translation is coupled to transcription in bacteria, and the formation of a terminator in the messenger RNA is based on whether or not the amino acid is present in the cell. So the presence of tryptophan affects the formation of the terminator, and in that way, if tryptophan is present, the terminator forms and transcription is stopped. And we also looked briefly at the SOS response, and as I've just described to you, there are multiple genes involved, and these multiple genes are repressed by a single protein, and this repressor protein is degraded in the presence of a signal such as DNA damage, which then leads to the activation or loss of repression of genes that respond to DNA damage. Okay, well, I hope this this presentation has been helpful to you. As I always say, it's important that you get your information directly from the textbook, but I've tried to work this material so that it's easier for you to flick through the textbook and make sense of the information that's there. Okay, so thank you for your time, and see you next week.